All right. So yeah, so what does it mean to be eukaryotic? Evolution of mitochondria and chloroplast again, and then how mitosis is different in some eukaryotes. So back to the fossils. So prokaryotes were the first eukaryotes. I mean, yes, did I say prokaryotes? I meant to say protists. Oh my gosh, this is going great. So protists were the first eukaryotes. But the exact sequence of when those events of like what does it mean to be eukaryotic is unknown. But we do know what those key events are, but we just don't know the exact timeline. So we do have some fossils, some microfossils that are 1.5 billion years old. We have found some microfossils that are even older than that, but we're not 100% sure that they are eukaryotes because they're chemical traces, but we don't have fossils to support how those chemicals were made. And then eukaryotes in Greek means true nucleus. So eu means true, and then kary actually means kernel or nut, but it's describing the nucleus. So we have to keep in mind that as eukaryotes were evolving, we have some horizontal gene transfer that's occurring, as well as the infolding of membranes and engulfing of other cells, and we're going to talk about that. So the nucleus and the ER arose from membrane enfoldings, and that's probably the first event because, you know, when they say, what's the first event that happened with eukaryotes? Well, eukaryotes have a nucleus and prokaryotes don't. So the first event had to be the formation of a nucleus, and so they believe it came from the infolding of the plasma membrane, because all cells have a plasma membrane that just kind of turned into the nucleus, as well as the endoplasmic reticulum. So, yeah. All right, moving on to the mitochondria and the chloroplast. So mitochondria evolved from engulfed aerobic bacteria. And I'm not really going to go in depth of endosymbiosis here because we have covered this numerous times already in this class. Chloroplast, kind of in the same boat, engulfed photosynthetic bacteria. The chloroplasts that were engulfed, they all think they came from a single line of a cyanobacteria of some type of species, but the organisms that engulfed them are not monophyletic. So it's just saying that it's happened independently in different types of organisms. So with endosymbiosis, we have some ancestral eukaryotic cell that has already had internal membranes, you know, a nucleus, an ER, and so then it engulfed some aerobic bacteria, and then that aerobic bacteria became mitochondria. With chloroplast, it originated in the same way, some eukaryotic cell that already had internal systems of membranes engulfs the photosynthetic bacteria. So we can also have, so that's primary endosymbiosis, but we can also have secondary endosymbiosis, where it engulfs another eukaryotic cell that has already undergone primary endosymbiosis. And one key feature of this is the number of membranes. So when you go through primary endosymbiosis, then you'll see a double membrane inside, some type of organelle that has a double membrane. When you go through secondary endosymbiosis, now how many membranes will be around that? 4. Yep. Okay, so over here I just have the endosymbiosis, the primary. Okay, well, this is kind of showing the cyanobacteria and the aerobic bacteria. So there's my mitochondria and my chloroplast, and now they live in symbiosis or symbiont inside this eukaryotic cell. And then here's primary endosymbiosis where it's already been engulfed, but then it gets engulfed again. And so now our number of membranes is 4. So we'll later on, I think, hopefully at the end of this lecture, we'll talk about brown algae and how they got their chloroplast, which is through secondary endosymbiosis. So the evidence for endosymbiosis, again, this is slight review. Both mitochondria and chloroplasts contain their own DNA, and that DNA that they have is very similar to bacteria in size and character. So it's smaller in the number of genes it has, and character just means that it's singular and circle. Eukaryotic DNA is multiple and linear, but mitochondria, it's prokaryotic. It's singular, and there's one of them. Okay. Mitochondria also have their own ribosomes. So when mitochondria want to reproduce, the cell doesn't make more mitochondria. The mitochondria make more of themselves. So they have ribosomes that do this. And those ribosomes are very similar to bacteria ribosomes, which can be kind of a problem. Like when you get sick and you have to take antibiotics, well, sometimes the antibiotics that you take target bacteria ribosomes. Well, mitochondria ribosomes are very similar to bacteria, so they could also be affected by that antibiotic. And then finally, I just said right here that they replicate by binary fission, not mitosis. And binary fission is how prokaryotes reproduce. Okay, the last thing about eukaryotes is that mitosis evolved in eukaryotes. So we needed a way to divide our chromosomes and make sure that cell contents gets divided evenly. So mitosis is the separation of those chromosomes with a nucleus, and cytokinesis is the division of the cytoplasm. Now there is one difference in mitosis with fungi and some protists. So in mitosis and prophase, the nuclear envelope starts to break down. But in fungi and protists, the nucleus doesn't break down and mitosis occurs within the nucleus. So when we get to fungi, which I think is chapter 31, we'll talk about this special mitosis that does not involve the nucleus breaking down. But in all other eukaryotes, the nuclear membrane does break down prior to mitosis or in prophase. All right, review questions. Oh, wait, everyone got this? Okay. So the evolution of which of the following provides for higher levels of genetic recombination and variation? B, sexual reproduction. The next question, the origin of which of the following does not represent an endosymbiotic event? C, the nucleus. Nucleus did not come from endosymbiosis, only the mitochondria and the chloroplast. Which of the following statements about Protist is incorrect? It is B. Protists are prokaryotic organisms. Nope, they're eukaryotic.
Is it because they have a nucleus?
Right, they have a nucleus. So protists are eukaryotic. Okay, so this statement is incorrect. We'll learn about these statements later on. Here we go. Overview of protists. So I'll talk about how do they classify protists, which is very interesting. And then we're going to go back to those 6 supergroups that were mentioned in the previous chapter, and I guess we're going to go in depth with them. And then we'll talk about how protists move, and there's 2 ways, flagella and pseudopods, and then talk about how they obtain energy. So phototrophs, phagotrophs, and osmotrophs. So What is a protist?
Is that all the slides you see on TikTok?
Wait for it. Wait for it. If a eukaryotic cell lacks features that would place it with fungi, plants, or animals, you just might be a protist. So the kingdom Protist, there's 4 kingdoms in the eukaryotic domain. Plants, okay, animals, protists, and fungi, okay. Protist, you know, if you're not fungi, plants, or animals, then they just say you're going to be a protist. So it's not monophyletic, okay. It's not, they don't share a common ancestor. It's paraphyletic. It's really technically not a kingdom. It's just used for our convenience to classify animals. All the other ones, you know, they have like a common ancestor, but this is like a bag that doesn't fit everything else. It's like just a mix mash of everything, okay? So protists, because they are this catch-all, you know, they are probably the most diverse kingdom in the domain Eukarya. So they can be unicellular, multicellular. We'll talk about colonial, so you'll see that a lot. They're microscopic. Some can be as huge as trees. Kelp is a protist. It's not a plant. You're like, what? Yeah, okay. So kelp, you know, they grow off the coast of Washington and Oregon, and they can be like over 100 meters long. Okay, so yeah. They have radial symmetry. They have bilateral symmetry. They have unique cell surfaces, and we'll talk about that coming up here. But really, the only unifying principle of a protist is that it's not a plant, animal, or fungi. That's it. So, yeah. So for when we look at life, a lot of it was prokaryotic, and then our protista somewhere appeared. And then from protista, we get to plants and all the other animals. But this tree is just paraphyletic. Okay, it's not monophyletic at all. There is debate about protists right now because they're like, well, you know what? Are protists best considered as several different kingdoms within? Because there are some protists that act like plants. There are some protists that act like animals. And there are some that are like kind of in between that can go back and forth. So they're like, well, maybe, you know, for the ones that act like plants, the ones that act like animals, the ones like, maybe they should all be separate kingdoms. But right now they're just going to, you know, put it all underneath protista or protist. You know, and then they're like, well, maybe this protist technically, maybe they do belong in the plant kingdom. So the protist, it's constantly changing. In fact, this edition is way different from the old edition, hence my scramble to make the new PowerPoint. Because I was like, oh, well, that's interesting because you grouped it with this before. So it's kind of cool that I'm seeing both sides of it and you don't see that. But yeah, it's changing. So anyways, eukaryotes are organized into these 6 supergroups that all contain protists. So we will go through these in the next section. So the Escavata. So you can kind of see that with this blue color here with the diplomons and the euglenoids and the Parabasalids. The Chromoaviolata. So this group over here, and you can see that they're subdivided into the aviolata and the Straminopelia. The Rhizaria, where are you guys? Yep, so there's 3 kind of major groups of Rhizarias, and we'll talk about what does it mean to be Rhizaria. Amoebozoa, really just one clade of those. And then this one I need to look up how to say, but the Opisthoconta, which includes your Dinoflagellates, and then fungi will be chapter 31, and then animals will be, I think, chapter 32 and 33. So, yeah. Oh, and I totally forgot to say Archaeoplastia. That's where plants fit under. But we'll talk about red algae and green algae. So, okay, so protists, of course, absolutely. Okay. So there are 2 kind of main way protists can move, and that is with a flagella. So with flagella, they use it to propel themselves. Cilia, they kind of put underneath this. Cilia are kind of tiny, like hair-like projections. It can be used for movement, but a lot of times it may be used to create water currents So that way food enters a certain part of the organism so it can feed. And then we have pseudopods, which are false feeds. So you see this in the amoeba, these extensions, cellular extensions. These are called pseudopods. In your book, in this new edition, they don't talk about the different pseudopods. In the old edition, they said there's 3 types of pseudopods. You could have like lobopodia, philopodia, and exopodia, and they all just talk about the shapes of the different podias. So, but they all do the same thing. They help with movement. They ooze out, and sometimes they adhere to surfaces to kind of roll their body in that direction. All right, moving on to nutritional strategies. So there are lots of ways protists get their food or obtain energy. So they can be heterotrophic, and we know what heterotrophic means. They get their organic molecules from other organisms, usually by eating them or consuming them. So phagotrophs fits underneath heterotrophic. So phagotroph just means that they're going to ingest food by pulling it in through a vesicle. It's almost like endocytosis, but they call these special vesicles either food vacuoles or phagosomes. Okay, protists can also be autotrophic, which means they make their own organic molecules or food. So photosynthesis or photosynthetic protists fit under here, as well as chemoautotrophic. So instead of using light, they use chemicals to make their organic molecule. And then down here, the third one is called a mixotroph, which means they both use light and they consume other organisms. So I'll talk about how they can switch back and forth based on the conditions that they're in. Okay, final slide for this section. Protists reproduce asexually or sexually. So for asexual reproduction, there's a few different ways they can do this. Mitosis, where it just splits and you have identical cells. Budding, where it buds off the parent, so the daughter cell starts out smaller, but then it can grow to the adult size. Schizogony. This one's actually kind of cool, and it's the picture down here. So we have cell division kind of like halts, okay, and we just make a bunch of nucleuses instead, so the nucleus divides. And then all of a sudden it will go through cytokinesis, and each one of these nucleuses will become individual, daughter cells. So there's multiple mitosis where we're just making multiple nucleuses, and then we'll divide the cytoplasm at the very, 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 very end simultaneously. And then they can also reproduce sexually. So meiosis is a major eukaryotic innovation. You know, we have the union of the haploid cells, and there's just this advantage of genetic recombination, genetic diversity with that. I guess if I wanted to add one more unique characteristic of eukaryotic organisms, I guess you could throw multicellularity in here, because with multicellularity you get cell specialization, but I don't know, I just felt like you'll see it later on in this lecture. So, all right, review question. Which of the following is correct regarding the phylogenetic nature of kingdom Protista? It is C. Yep, the entire kingdom is paraphyletic. They have not decided to do this yet, okay? So that eliminates that. And they are very diverse. They don't really share characteristics other than that they're not plants, animals, or fungi. So C, paraphyletic. Okay, moving on to Escavata. Escavata, yeah. So main features between some major groups underneath Escavata, which are the diplomonads, Parabasalids. I'll talk about euglenoids and kinetoplastids. Holy smokes, lots of vocab in this one. New terms. All right, so the unifying feature of Escavata is this unique feed-in groove that it has. And you can kind of see that in these pictures right here, and in this cartoon picture right here, and this cartoon picture right here. They're also unicellular, one-celled organisms. So there are three distinct groups underneath Escavata: the diplomonads, the Parabasalids, and the next slide I have the Euglenozolens. All three of these big groups are similar to each other due to cytoskeletal structures as well as DNA sequencing. So first up, diplomonads. They lack a functional mitochondria, so they think they gained it and then lost it. They also have multiple posterior flagella, but the thing that really sets diplomonads apart is they have 2 haploid nuclei per cell. So you can definitely see the 2 nuclei in this picture right here. An example of a diplomonad is, I'm going to butcher this name, but Guyardia Intestinalis, which is a parasite that can be passed from human to human through contaminated water, and it causes diarrhea. One of the most important hosts for diplomonads is the beaver. So then, the beaver lives in the water, and yeah, beavers poop, and then it gets in the water, and then humans may drink that contaminated water, and then they get diarrhea. Parabasalids, an undulating membrane used for locomotion. So you can kind of see this fold right here. They're kind of like ribbon worms, and they just kind of like flutter through their environments. Semi-functional mitochondria, 1 nucleus per cell, and an example of that is Trichomonas vaginalis, which is a STD. So, yeah. There's another type of Parabasalids that actually has a symbiotic relationship with termites. That's how termites are able to digest cellulose or wood. Okay, continuing on, the third big group is the Euglenozolens. And these guys are probably the earliest to possess a mitochondria and not lose it. But they are characterized by some type of anterior pocket or chamber from which 1 or 2 flagella will emerge. So you can see here's this pocket, and here's one big flagella, And then here they have a reduced flagella. They don't have a cell wall. Instead, they have these strips of protein called a pelysyl. There are many different species of euglenoids or the euglenids, but a third of them have chloroplast and are autotrophic. And if you place them in a dark environment, then they become heterotrophic. So they can kind of flip-flop. No sexual reproduction has been observed in euglenoids. So those are the top two pictures here, euglenoids. Another group underneath the Euglenozolens is the kinetoplastids. These guys are unique in that they have a single mitochondria in each cell. And this mitochondria has like two types of DNA, a mini circle and a maxi circle. Okay, there is a difference between the mini circles and the maxi circles. The maxi circle is needed for protein production, and then I think the mini circle is needed for making mRNA. So, yeah. Okay, now the kinetoplastids are actually responsible for a lot of diseases, and we call them trypanosomes. So disease-causing kinetoplastids are called trypanosomes, and these are three examples. First one's called African sleeping disease, which can cause extreme lethargy and fatigue, and it's passed on by a vector, the tsetse fly. Another type of trypanosome, the, Leishmaniasis. This one's transmitted by another fly, but it's called sandflies, and it causes skin sores and damage to your internal organs. And then finally, chagas disease can lead to severe cardiac and digestive problems. So these guys are like parasites, and they cause these diseases. So controlling trypanosomes, there's few different ways that we've attacked it. We release sterilized tsetse flies, so that way they can't reproduce. It kind of wipes out a generation. They set traps scented like cows, and then these traps are heavily doused with insecticides, and that takes care of a lot of them. But sequencing all three of these genomes has revealed that they have a lot of common genes, and so they're hoping to come up with a cure or a vaccine that will just take care of all three of these. It has been kind of tough to make a vaccine because these trypanosomes, they change their protective coat on the outside. They have so many genes to choose from. So the antibodies that we create for vaccines, they won't bind to them and identify them as foreign because they have this ability to change like their identity or their ID card. All right, can I move on?
Wait, hold on.
Yep, that's fine. All right, which protist that lacks mitochondria would most likely be classified with the, or a protist, wow, I read that question wrong. A protist that lacks a mitochondria would most likely be classified with the, yeah, they're the closest, Parabasalids. So you're probably looking for the diplomonads. You're like, that's not one of the options, but Parabasalids are, you know, it says, then we have a note that says like they have a semi-functional mitochondria. Yeah. Which of the following statements best describes the term kinetoplasmid? It's a organism with a single mitochondria in each cell. We haven't even talked about aviolata, so that took out B and D. And yeah. Okay, another one. Why is it so hard to develop a vaccine against chagas disease? Read all of them. I know that one seems real good, but there's a better one. It's C. Yeah. Yeah. Okay, fourth section. Secondary endosymbiosis in the chrome aviolata. So what are some members? What's the distinguishing feature of them? The function of something called the apical complex and apicomplexans. And then we'll talk about straminopeles and oomycetes or oomycetes, depending on how you want to say it. So here we go. So there are two branches underneath the chroma aviolata. We have the aviolites and the straminopeles. So the aviolites will include the dinoflagellates, the apicomplexians, and the ciliates. The straminopeles include the brown algae, diatoms, and the oomycetes, which is water molds. So the common trait for the aviolites, they have these flattened vesicles, and that's a typo there, flattened vesicles called alveoli, just underneath the plasma membrane, and it's used in transportation. The common trait for straminopeles are fine hairs found on the flagella, so their flagella is hairy. So the next slides, I'll talk about dinoflagellates in depth, apicomplexians in depth, ciliates, brown algae, diatoms, and the oomycetes. Okay, so first up, dinoflagellates. Okay, so dinoflagellates are photosynthetic, one-celled organisms that have two flagellas, as you can see in this picture right here. They are found in marine as well as freshwater environments. Some are luminous, and I'll show you a picture of one species on the next slide. Their structure, you can kind of see that they have these plates put it together, well, kind of pieced together, and they're made of silica, and they have these grooves, and that's where the flagella is housed. And the word dino actually means whirling. So these flagella, they spin, and these organisms, how they move in the water is they whirl. They just kind of spin like a ballerina in the water. One thing about dinoflagellates that are very important for us to study is that they undergo blooms or red tides. So they go through explosive growths when conditions are right. And you can kind of see in this picture here, this brownish kind of pinkish color water. And the reason why they're so harmful to our economy as well as wildlife is that they produce toxins that, it says in your book here, toxins that inhibit the diaphragm, causing respiratory failure in many vertebrates. So fish die, birds that land in the water, and if they get enough of them inside their body or absorb that toxin, birds die, so it can wipe out a lot of wildlife. Okay. Dinos, because they are photosynthetic, they do have a chloroplast or a chlorophyll, and so they got that probably from endosymbiosis. They can reproduce sexually, but they mostly prefer asexual reproduction. And then another weird thing about dinoflagellates, when we sequence their DNA, they don't have any proteins in their DNA or histones, and they honestly don't know how that occurred. Because like all eukaryotic DNA have proteins that the DNA wraps around so it can supercoil and be packaged properly. But here, there's no histones. They lack histones. So they are the only eukaryotic organisms that don't have histones. And they're like, what? How is that possible? So here are some more examples of the dinoflagellates. This is the one, like this picture right here. That's that species in a cartoon diagram. This one right here lacks a cellulose armor. As you can see, all of these kind of have like heavy-duty armor on them, but this one does not. And it's just one type of species that's bioluminescence, which can glow. And if you look at the name, Noctilucia, nocturnal, night, light, luca. Okay, so night light. Yeah. All right, moving on to another group underneath the aviolites, Amphicomplexians.
please have your attention for a quick announcement. Snowball royalty, please head to Mrs. Steele's room at the end of